Well, thank you, Karen. Thank you, all of you, for being here. A special thanks to the David family, to Gabriele, to Ariel. It is just a fantastic honor to participate in this great Dan David Prize that you have created, which will stand as an eternal legacy to a fabulous man and a really great vision. And it's a great honor to be part of this chain that has gone for 11 years and will go far into the future. So, David has talked a bunch about yeast, and yeast is a model system. A model looking to the human, particularly cancer he's referred to. Craig has spoken eloquently about the power of reading and writing bacterial systems in just huge numbers. I would like to turn in the closing talk of this symposium before we have our round table about the human genome itself, what we've learned about the human genome. And in particular, what I'd like to do is, I think very much as Karen said, talk to the students. I'm delighted to see so many students here because I want to give a sense of the rate of change of science. How, to me, what's so fabulous about genomics and what's so hard to understand as a student who comes into a field and is sort of learning where the field is at today is to also learn the first derivative, and in fields like genomics, the second derivative of the field, how rapidly things can change over time. And so my theme today is going to be to talk about what we have learned over the past 10 years, how we've learned some of it, and where it's all going, to give a sense of that. So I've called it Secrets of the Human Genome, to emphasize that it's mostly about things we didn't know about 10 years ago and didn't really even suspect 10 years ago. And the goal is not to say, wow, look at all the things that were learned in the next 10 years, but to say to the students how much more there is to learn in the coming 10 years, and think not linearly, but think exponentially in terms of how that will go. So, a word about the Human Genome Project. All of us here today, David, Craig, I have played roles in this Human Genome Project, and just a very brief word about it. It was the first time that the biological community came together around a real goal-directed project. It was really in the mid-1980s that this idea of, yeah, David laughs, he recognizes, yes, there's a picture he and I have shared for 2 decades. It came together in the mid-1980s with the crazy notion that one could sequence the human genome, and it was certifiably nuts at the time. There were all sorts of reasons why, take a 100,000-fold scale up from what you could do, and there were arguments about should we do it immediately and leap into it, or should we try to lay groundwork in different ways? And the scientific community came together and talked and debated and agonized for several years before setting out on what was a pretty wise plan. To lay first the genetic map of signposts up and down the genome, a physical map, being able to show you could get all the intermediate DNA, then read out sequence, and then annotate it with regard to the genes that were there. And very importantly, the idea that all that information had to be made freely, completely, immediately available so it could be used by people everywhere in the world, in industry, in academia, in every country with no limitations, and mixed and mashed together. So that was the idea. It involved changing all sorts of things. It involved Craig showing you his Henry Fordish slide. This is kind of our Henry Fordish slide of what it felt like in the height of the Human Genome Project with lots of robots moving around and conveyor belts and all sorts of things going on. It was an interesting time because it really involved changing the way we thought about doing biology. It also involved changing the way we thought about collaboration. The Human Genome Project depended tremendously on international collaboration over the course of about 13 years from 6 different countries, the United States, the United Kingdom, France, Germany, Japan, China, 20 different labs, all working together, some very large, some small, but toward this common goal. So give or take, the Human Genome Project was completed many times. It was completed, we really liked celebrating. And so there was the announcement of the completion, the publication, then a finished sequence, and the publication. This gives us great occasion about now to celebrate the 10th anniversary many times, and we're going to be doing that as well. It's all good fun in that sense. But what I want to talk about is not those days. I want to talk about what's happened since. There was a certain sense that all this crazy mapping and sequencing and scale and all this stuff, we'd finally be done with it after the Human Genome Project, and we could go back to business as usual. There was, I think, a strong desire on the part of many to have that be the case. But it turned out that there were really permanent changes to the way we approached biology. One was the idea of being able to build comprehensive pictures, comprehensive maps, and learn from them. Having one sequence of the human genome made it possible to build many more maps on top of it, because it became a scaffold for assembling fragmentary information. And so we went from this simple original notion of genetic maps, physical maps, sequence maps, to building gene maps on top of the sequence, maps of evolutionary conservation across species and millions of years, maps of chromatin states across that sequence, maps of three-dimensional structure across that sequence, laying fragmentary information to build these maps. With regard to disease, maps of inherited genetic variation, specific disease associations, evolutionary selection in the last 5,000 to 10,000 years, maps of cancer genes, all these sorts of things, these powerful tools became more powerful because you could cross-reference them. When you found a disease association, you could see it sat above some chromatin mark and some evolutionary conservation, and begin to build that. I mean, to us it's now obvious with Google Maps, layering all the annotations on Google Maps, but back then it wasn't so obvious that that was what it would be. The other thing that was distinctive in this era was completeness gave us the ability to detect things based on little fragmentary signatures. You could do gene expression because if you took a little fragment of DNA and used it as a probe, you would know it was the only gene that had that fragment of DNA. And if you flew proteins in a mass spec and you saw a particular amino acid sequence, you could know that that was diagnostic of a particular protein because there were no other choices, because you had essential completeness. So those were really important ideas. Mapping, cataloging, gave us a great deal of power to do experimental biology. So what have we learned? Well, the things I'll just try to talk about today, and I'll talk lightly about some of them and maybe in depth about two of them. What have we learned in the last 10 years about genome sequencing? What have we learned about the functional elements that are encoded in the genome? About the evolution of the genome? About the basis of inherited disease? About the basis of cancer? And about human history? So I'm going to talk about all of them, but I'll probably say a fair amount about some functional elements in the genome that I'm very fond of right now, and a fair amount about the basis of inherited disease. And I've just picked those two because of time, but I'll talk about all of those in some way. So the new world of DNA sequencing is just mind-boggling. What has happened to DNA sequencing since we were done with what we thought was large-scale DNA sequencing is truly stunning. The view from 2001, when the first paper on the mapping and the sequencing of the human genome came out, before that, there were 4 eukaryotes that were sequenced, 38 prokaryotes, and all told they added up to about 500 million bases of DNA. Well, today we've got hundreds and hundreds of eukaryotic species. We've got thousands and thousands of bacterial species, completely done. As Craig has said, we've got vast metagenomic samples that point to hundreds of thousands of different species, depending on how you count. We've got thousands of human whole genomes, tens of thousands of human whole exomes, and people are talking about getting complete sequencing across tens of thousands of vertebrate sequences. That's in 10 years that picture has changed. And it's changed, of course, because of continuing growth in technology. The machine on the left, give or take, was the workhorse of the Human Genome Project, and all sorts of machines based on the principle not of using 96 capillaries, but using 1 billion spots on a slide and looking at them optically have accelerated things dramatically. The feeling for us at the Broad Institute was something like this. We were proud as punch in 1999 to sequence 1 billion bases of DNA, and then over the course of the coming years to get that up to 70 billion bases of DNA per year. being produced. We thought this was really wonderful. As the new technologies came in, the next 2 years looked like this. The next year after that looks like that. The year after that, it just runs off the slide. You know, it's up to from 70 to, you know, let's see, where was it? 70 to about 1,800 a year, to about 20,000 billion bases, to, you know, 300,000 billion bases, and that's just our shop. This is what's going on in the world. More interestingly, as a general number, is the cost curve, which has dropped like this. The folks in the semiconductor industry are so proud of Moore's law, things falling exponentially over time with a certain constant halving every 18 months or so. This is the drop that we've experienced since about 1999 in the cost of sequencing, getting close to about 1 million-fold. If you go back to the early days of the Human Genome Project, the cumulative drop is way in excess of 100 million-fold over that period, but just almost 1 million-fold in the last 12, 13 years or so. Really quite wonderful. And what are the implications of that? Well, the implications are that sequence has become the general tool for discovery, a general readout for any kind of molecular problem, any kind of molecular interaction, any species question, population, because it's cheap. If you can figure out how to reformulate a question, a biochemical question, a three-dimensional question, as a sequence question, you can get answers out very quickly. So increasingly, we just think about how to turn things into a sequence problem. Sequencing is also going to become, but I'm cautious about how it becomes, a routine tool in medicine. You know, not willy-nilly out sequencing everybody's genome, but for good reasons, doing germline sequencing, cancer sequencing, immune repertoire sequencing, microbial sequencing. And this will be driven as the benefits and the cost cross over. Sequencing will need to hit $1,000 a genome. We're getting close to that. We'll get there, you know, not so long. I think it'll need to hit $100, and I think there are good ideas now for getting it to $100. And below that, you start getting less than the cost of drawing the blood out in the medical system. And so it's not clear that the incentives get to drive it a lot less, but there probably will be clever ways to go beyond that. There are even, you know, those are cute devices that don't actually work yet, but are conceptually devices that might get you to costs like $100 perhaps. All right, so that's technology. Technology has been mind-boggling over the course of the past decade. But what about understanding the genome? What's in the human genome? Well, what was the view from 2001 about what was in the human genome? Now, I teach freshman biology at MIT, and I have done so for 20 years, and every year I tell the students what we know. What we know in particular about the human genome in one of the lectures. And the things that I could confidently tell them in 2001 were things like the following. The number of protein-coding genes in the human genome was somewhere between 35,000 and 120,000, because there were two papers back-to-back in Nature Genetics that reported those two numbers, bracketing the number of genes. We could draw a picture that said the vast majority of the information was protein coding. There was some regulatory information in the human genome, but the vast majority of it was, you drew a picture with some exons, a little bit of promoter up there. We knew that there were a few funky, strange, non-coding RNAs, but they were clearly a minority population, a handful of examples, belonging to some classes, and about 7 other weird examples. And we knew, as has been referred to already, about half of the human DNA is transposons, and that they were junk, just detritus, a burden on the genome. So the progress in the past 10 years is that we now know that all of those statements are wrong. That's progress. And how do we come to know these things? How do we come to know that all of that is completely wrong? Well, one of the most powerful tools is evolutionary conservation. Now, you would say, if I want to figure out what a nucleotide does in the human genome, change it and see what effect it has. Go in, alter the nucleotide, grow up another human being with that altered nucleotide, and measure the phenotype of that human being. Well, as you will imagine, that has some issues. There are practical issues in the time needed to do that experiment, and ethical issues involved in doing such things, so that it's not really practical. But of course, if we thought about it in advance and had arranged this experiment much earlier, we could be changing the nucleotides, collecting the data, and of course that's what evolution does. The ancestral mammal 100 million years ago had a mammalian genome, and then mutations occurred and they piled up and they got selected against or they got tolerated. And by comparing mammalian genomes today, we can see what matters, what doesn't matter so much, at least with regard to things that work across all mammals. So, of course, as soon as we had a human genome, the community wanted very much to have a mouse genome, so we got a mouse genome. Once there was a mouse genome, there was a lot of interest to get a rat and a dog genome. So we got a rat and dog genome. And as so much got learned from the mouse and the rat and the dog, there was pressure to collect, and very good scientific reason, to collect more and more genomes. And now there's something like 48 mammalian genomes that have been sequenced and are freely available on the web. And what have they taught us? Well, one of the things they taught us was that there are a lot fewer genes than we thought. In fact, the gene count is not 100,000 or so. It's about 21,000 protein-coding genes in the human genome. And that's held up remarkably well over the past couple. And people discover a couple new genes from time to time, but there's not a lot of them. So, although there are a lot fewer protein-coding genes than we thought, what's very interesting is there's a lot more important conserved information than we thought. It's just not in protein-coding genes. So in fact, you can do a little bit of mathematics here and look at the distribution of conservation of nucleotides across the genome. And you get a curve of how much conservation there is. And you'll notice that blue curve has a fat tail on the right, indicating more conservation than you might expect if it was just a random symmetric chance process. You can take transposons, which are mostly dead, and use them as a background distribution, and they show a nice symmetric red distribution. And by subtracting one from the other, you can draw an inference, which is again held up remarkably well over the course of recent years, that about 6% of the human genome encodes evolutionarily conserved information. Evolution has been lovingly conserving this information, but only about 1.2% of it is protein encoding. So the majority of what evolution has bothered to conserve is not protein coding. Now, at the beginning, we had to do this by statistical inference, looking at those curves, because in fact, we could only detect in specific cases about 500,000 of the elements that were being conserved, about 1.6% of the genome, and they had to be pretty big to detect. But more recently, in work involving 29 mammalian genomes, it's now possible to find about 3 million of those conserved elements, representing about 4.7% of the genome. So we can put our fingers on large numbers of these conserved elements. What are they? Many of them are pretty clearly turning out to be regulatory elements, cis-regulatory elements. And it's very interesting to ask where they live in the genome and what they tell us. So you could take the very most conserved of those cis-regulatory elements and ask, for example, where do they live in the genome? And it turns out they, which are shown here in yellow, have a different distribution than the density of protein-coding genes. The most dense regions in protein-coding genes are not the most dense in these non-coding elements. Turns out these non-coding elements preferentially live in gene-poor regions of the human genome, but not regions with no genes, just few genes. And which genes are there are fascinating. It turns out that many important early developmental genes live in these regions. They live there, many transcription factors from early development, axon guidance receptors, and it's not a surprise to see what's going on. I'll give you one example here, the SATB1 gene. There, the protein coding region is in red, and all of this other stuff that lives around it is in purple. And what you can see is a tremendous amount of ancillary regulatory sequence around this one gene to get this early developmental transcription factor to be expressed correctly. So it's given us a very different picture of the regulatory controls across the human genome. Now, an interesting question is, how's this stuff changing over time? How's the invention process going? Well, one of the disappointing facts about proteins in the human genome is that we have not been very creative in mammals. We have not invented a lot of new protein coding sequences in the course of the last 100 million years. Most of them are shared across all of these mammals. In fact, it's really pretty derivative. You wouldn't get high points for creativity. Most of the invention turns out to be in these purple things. these regulatory regions, and you can see that by sequencing the marsupials. So if you sequence marsupials there, and you compare the marsupials who diverged away from the placental mammals 90 million years before the mammalian radiation, you can compare what's common amongst all the placental mammals, but is not shared by these marsupials, and you can figure out how much was invented in those 90 million years between 180 and 90. And you can find out that about 16% of what we have shared amongst placental mammals was invented during that period. It lets you set a clock on that invention. We're inventing about 4 new nucleotides per year. That's the rate. You know, what you can also find is the vast majority of it is not protein coding. It's regulatory sequence. So then you got to ask, how are you inventing all this regulatory stuff? What do you know, where's it coming from? Suppose I want to regulate 15 genes coordinately, and I want to get them to be coordinately regulated with each other. Do I have to evolve separately the regulatory controls in 15 places in the genome? That's going to be hard to come up with a complex regulator in 15 places, perhaps. But maybe there's a better way. Maybe I invent a regulatory sequence in one place, and it works really well, and then I just distribute it around the genome. And so a complex element can show up in many places around the genome rather quickly. And if you thought about that, you'd say, well, great, transposons could carry things around the genome. Wouldn't that be a good thing to put them to use for? You know, they're parasites on us, but maybe we can get them to do some work for us. Well, in fact, it turns out to be the case. It's clear now, by comparing these sequences, that at least 18% of the newly invented stuff lives in the remnants of transposons that carried it around the genome. And 18% must be a gross underestimate because much of the remnants have degenerated and we can't see all of them there. So it looks like transposons, while they are parasites, while they are a burden on the genome, they do seem to be an important part of disseminating evolutionary innovation, which is kind of cool. So these are interesting pictures about what we're learning about the human genome. Now, other things that we're learning about the human genome, about what functional elements come not so much from evolutionary comparisons, but they come from epigenomic studies, the marks that sit on top of the human genome. And so what was particularly fun about having a sequence of the human genome and then these new tools for sequencing is we could start building chromatin maps. We could take any particular modification of the chromatin proteins around which the DNA is wrapped, and those chromatin proteins have scores of different modifications on them, and if you have an antibody against a particular modification, you can pull down the histones that have that modification. They drag along the DNA that's there, and you sequence that DNA and see what DNA is living in places that have this histone modification. And you can, if you want to look at green modifications, you can get the distribution of the green modifications, and you can see if you're looking at red modifications, where the DNA is that, where the red modifications are located there. And you can build these comparative maps of histone states, epigenomic states across the genome. So we started doing this, and many folks started doing this, and what was interesting is you could pick up patterns, and I'll just tell you a graduate student story here. There was this pattern that's found on an actively transcribed gene. It has at the promoter of this actively transcribed gene a big green mark, histone 4, lysine 4 trimethylation, histone 3, lysine 4 trimethylation at the promoter, and lysine 36 trimethylation along the transcribed region. So an actively transcribed protein coding gene has a green mark and a blue mark defined there. And so when a graduate student, Mitch Gutman, started in the lab, Mitch's assignment was take that mark and scan the genome and see if we've missed any protein coding genes. Were there any other places that had the green-blue mark? So he went out and he looked. That was the assignment, and he marked it up and he said, aha, there's a new one there. But he came back after a couple weeks and he said, I found a few thousand new ones. This was very disturbing because we had previously published the paper just the previous year indicating that we were largely done discovering new protein coding genes. And so, you know, there's a different person in the lab, and you had to figure out what did this mean. So there was a happy resolution here, which is he discovered a lot of actively transcribed genes, but they're not protein coding. What has become clear is that they are functional non-coding RNAs. We call them large intergenic non-coding RNAs or link RNAs. And I'll just tell you a little bit about these link RNAs because they're a very active, fun subject. And they're one of these surprises, one of these secrets of the human genome. Now, these large non-coding RNAs, as I indicated before, there were some examples known of these things, limited numbers of examples. Indeed, Shirley Tillman found H19, a non-coding RNA. And there were other non-coding RNAs. Exist, a non-coding RNA important in shutting down one of the two X chromosomes in females. But around the time of the publication of the Human Genome Project, there was about 8 or 9 such examples. Each had a different function, and it was really unclear what to make of them. And here we seem to find thousands of them. So what's going on? So to make a long story short, we took the link RNAs, we did gene expression to find out where they're expressed across a whole bunch of different tissues. We correlated them with where classes of protein coding genes were expressed and said, aha, this set of link RNAs is correlated with a bunch of genes involved in immune responses, and these guys are in brain processes, and these guys correlate with cell cycle regulation, etc. So that gave you a sense that classes of these link RNAs were related to important processes. Related in the sense of correlated with. But correlated doesn't mean functionally related. It doesn't indicate that it's a biological function. The only way you can do that if you're a geneticist, or frankly, if you're anything, is you gotta knock it out and see what effects it has. And so, over the course of the past couple of years, we've taken this very surprising class and begun to look at them. And we've chosen to do it in embryonic stem cells. Embryonic stem cells are a good choice because you can differentiate them into different pathways. They have well-characterized transcriptional networks. You know which transcriptional factors are in charge there. And they have well-characterized chromatin regulation. So, link RNAs, the first thing we ended up doing here was knocking out a bunch of them. Knocking out, there are about 200 link RNAs in these embryonic stem cells, and when you knock them out, you find that the vast majority of them have major effects on gene expression in the cell. Whoops, let's go back here. Vast majority have major effects on gene expression in the cell. At least 24 of them turn out to be essential for maintaining the pluripotency. That's a pretty important function. And about 24 of them are essential for keeping the embryonic stem cell from differentiating down a particular pathway. So these are pretty important functions. Now we can say they're functional. So then, okay, check, maintain the pluripotent state, repress specific differentiation. There's real functions, at least, we can ascribe to them. Now, how do they fit into this machinery? Well, it turns out that you can look at the genes that encode the link RNAs at their own promoters, and sure enough, the key transcription factors that run embryonic stem cells are present at those promoters. If you knock down those key transcription factors, you affect the link RNAs, and those genes get downregulated upon differentiation out of the ES cell state. So we know that they're installed in that transcriptional machinery there. Then what are they doing? Well, this turned out to be really interesting and surprising. These RNAs seem to be involved in binding to a wide variety of chromatin regulatory proteins, chromatin readers and writers and erasers, and they appear to interact each with multiple distinct proteins. Oops, go back. Multiple distinct proteins there. And it's clear that the proteins that they are interacting with, if you knock out the protein or you knock out the link RNAs, have similar effects, which tells us that they're actually genetically functioning together. It's actually a little bit like David's double knockout interaction experiments. It's the moral equivalent of that. And so what that tells us is these link RNAs bind to diverse chromatin proteins, and they clearly regulate common target genes. The best model we have for what's going on is that the link RNAs are flexible scaffolds that help assemble protein complexes. They organize a party. They have regions that bind to protein A, B, and C, and because they bind well to protein A, B, and C, they're now in close proximity to each other and can make complexes. This is known to be the case for the telomerase RNA that acts as a flexible scaffold, and our sense is that this may be the case for large numbers of RNAs that act as flexible scaffolds there. And the great thing is, instead of inventing a lot of new protein-protein interactions, you run off a new nucleic acid, and this new nucleic acid is able then to have the binding sites, and then much weaker interactions between the proteins will suffice because you've tethered them together. So the idea is, in different cell conditions, I make different link RNAs, and they help assemble different protein complexes. That's the idea you should take away, and you should also take away that we haven't proven any of that, right? That's a hypothesis, and it requires proof. And so an awful lot of work, that's a very nice idea. I hope it turns out to be true. What I've told you up to now is true. This is a hypothesis. And what we're doing right now is defining all the sites at which the RNAs interact with different proteins. You can do that in high throughput parallel fashion. We're defining, using other techniques, where the link RNAs bind in the genome and how they work in circuitry. So there's tons more. But my main take home is we thought we knew what was in the human genome, at least what the major classes were, and there were whole classes we didn't even know about in the human genome. So what's ahead? Well, there's a lot of work still to go. It's fun to just do quick sequences of everything, collect stuff, but the real work is comprehensive catalogs of all the transcripts and know what they do, comprehensive definitions of all the chromatin states, comprehensive definitions of the long-range genomic interactions, all the interactions between DNA and protein and RNA, and it's going to take this next generation to come along and do that right, because getting that stuff carefully as we got the sequence of the human genome, is going to be essential to meaningful interpretation. And then beyond it, we need to be able to understand the grammar of DNA sequence. Embarrassingly, we can tell you this region is an enhancer, but we can't tell you the grammar of an enhancer. Go write your own enhancer. We can write a bacterial chromosome, but to write a new enhancer for a new function, people still can't really do that. And that's embarrassing. It's a poverty of knowledge of what it takes to make that thing. And I think there's a lot of work right now using the tools of synthetic biology that will let us begin to define observation and synthesis. Analysis and synthesis will together get us to the point where we understand the grammar of regulation. All right, so that's kind of what's in the genome, what we've learned from evolution. Let me turn to disease. I want to say a little bit more about disease. So I believe we told the American Congress that one of the reasons for sequencing the human genome was that it would teach us about human disease. So how's that going? Well, it's always good to check. So Mendelian traits, simple Mendelian diseases. There were 4,000 plus Mendelian diseases that were known in this wonderful catalog that Victor McCusick put together, the Mendelian Inheritance in Man catalog. And actually, it's gone quite well. Just before the Human Genome Project started, there were only about 70 for which the molecular basis had been identified through molecular cloning. By the time of the publication of the Human Genome Project papers, there was about 1,300 that had been pinned down to a gene. And then with the power of all this human genomics, the genomic sequence, it's gotten up to something like 3,200, depending on exactly how you count. And in fact, that's the majority. Right now, there's only about 1,800 still to go. Maybe that's even falling further. In the case of a simple recessive disease where you're guaranteed you're going to get a knockout on each of 2 chromosomes, you can almost do it from a single patient, although a few is good. For a dominant disorder, it's harder, but people are beginning to pull together a lot of work, and I think we'll see this fall pretty quickly. How are we doing, though, on the more common complex stuff? The stuff that most of us will suffer from? Well, at the time of the Human Genome Project, starting in 1990, the number of loci in the human genome that we knew really affected the common diseases was about 1, the HLA locus. By the time of the Human Genome Project papers, it was about 24, not so much. But in the mid-90s, a number of us had this notion that one approach to this might be to look at the variation that we could define in the human population, common genetic variations, common genetic variants, and some calculations said, depending on what you called common, there might be 10 million, 20 million, somewhere in that ballpark, common genetic variations, depends where you set the cutoff. And if we knew them all, and we could measure their frequencies in people with a given disease and without a given disease, we could see which ones were enriched in people with disease and figure out which genes might play a role. Now, this was also certifiably nuts because at the time we only knew a couple thousand genetic variations, not millions, and the thought of testing tens of millions of variants in tens of thousands of people is hundreds of billions of genotypes. They were done by graduate students one at a time, and the graduate students on the whole objected to this concept. So, but happily things progressed. And even though we only had a couple thousand, within a few years, there was really actually in a parallel paper, a companion paper to the public Human Genome Project, was a report of 1.4 million polymorphisms in the human genome. That's now grown to more than 20 million of the variations that have been known. It's turned out that we now know they're locally correlated with each other, so you don't even need to use all of them because they're so well locally correlated that if you have some, you can predict many of the rest around them, and an international project worked out what's called the haplotype structure across the genome. And genotyping went from 1 at a time to 10 at a time, to 100 at a time, to 1 million at a time, to now about 5 million at a time. And now with sequencing, it'll get even easier. And so the experience of looking for common genetic variations that affect common diseases looked something like this. The average was about 1 new discovery across the entire world each year. It was exciting, but it was 1 per year. So in 2005, it was exciting because 4 discoveries were made. Then in 2006, you get 8 discoveries made. Then in 2007, when all the tools were now there and the maps were there, you get that. That's what 2007 felt like. And today, there's more than 1,500 such associations across more than 225 different traits. So it's quite a remarkable picture, but what are we learning from this? So let me take lipids as an example. From lipids, Mendelian genetics, rare genetic diseases, had identified a bunch of loci that cause Mendelian dyslipidemic syndromes, and some of them actually were drug targets. Well, what was very interesting was using the common variant approaches, looking across the genome, that list jumped to more than 100 loci that affect lipid levels, LDL, HDL, and triglycerides. Now, some of those have been really interesting from a medical point of view, and I'll just give some examples. This is Seik Kathresan, who did the study. He's at the Mass General Hospital and the Broad Institute. One of them, sortilin-1, a locus involved in a certain type of multi-receptor sorting. And this locus here is quite remarkable. The alleles of the locus decrease your LDL cholesterol by about 16% and lower risk of coronary artery disease by about 40%. And you make the mouse overexpressions and knockouts, and they have comparable effects, and you can really study these things very well. Another beautiful example that emerged from the human genetics is PCSK9. There are null alleles running around at about 2-3%, low-frequency common variants in the human population, and they're associated with quite substantial decreases, again, in LDL levels and in coronary heart disease. And what's great is now a number of pharmaceutical companies have made antibodies against PCSK9, and they have dramatic lipid-lowering effects. So this has clearly made its way into the clinic pretty quickly. Here's one that has real bearing on the clinic, but in the opposite direction. All of you must know, at least in the United States, everyone has been trained to know that there's a good cholesterol and a bad cholesterol. The good cholesterol is LDL and the bad cholesterol is HDL. And that's because high LDL levels is associated with heart attack, and low and high HDL levels give you protection from heart attack. Low HDL is associated with heart attack. So clearly it must be flamingly obvious that making a drug that will increase your HDL will protect you from heart attack. But of course, it's not obvious at all. This is just an epidemiological correlation that people who happen to have high HDL are protected. It could be that high HDL is a correlate of what's really protecting you. And just merely increasing the HDL isn't going to help. You have to deal with the thing that was giving rise to the high HDL. But nonetheless, on the strength of the epidemiology, 4 pharmaceutical companies have each invested $1 billion to make drugs that increase HDL. Well, one had a colossal failure. A second just had a colossal failure a couple weeks ago. And it's pretty obvious now why they had a colossal failure, because I told you we have all these genes associated with lipid levels. Suppose we take all the people who have the alleles that predispose you to high HDL and theoretically should have been protected. How'd they do on heart attack? Well, we got 100,000 people like that that have been looked at, and we could do the same with LDL. Well, in a beautiful paper that Sake and other collaborators have just published in the Lancet here, what you have is a really remarkable study that says that LDL lowering alleles protect you if you inherited them genetically. HDL raising alleles offer no protection whatsoever. And so, in fact, there's a perfectly good reason that this therapeutic hypothesis is wrong, and the genetics could tell you that. There are many other fun examples. I'll run through them quickly. Inflammatory bowel disease, there's now more than 100 loci, and they spectacularly fall out into 7 different pathways, telling us that autophagy plays a role in this disease, IL-23 signaling, innate immunity. There are beautiful mouse models that have been created that reproduce the human defects in colon crypts. It's really a very gorgeous example. So there are many, many things that have come out of this macular degeneration we now know as a disease of the alternative complement pathway. Type 2 diabetes. None of the genes involved in risk of type 2 diabetes have much to do with glucose regulation, it turns out. That's a pretty important finding. Some do have to do with things like zinc transporters and beta cells. Sickle cell anemia. Best way to protect a patient with sickle cell anemia would be to increase their fetal hemoglobin. Well, nobody knew how to do that for 25 years, but there's natural genetic variation in fetal hemoglobin, and those loci have been mapped, and when you interfere with those loci, they can dramatically increase fetal hemoglobin levels. Schizophrenia, we used to have few examples of genes in it, but now there's multiple genes involved in L-type calcium channels, 5 genes involved in the miR-137 pathway, and we're getting light shed on these different diseases. So I'm going to skip over that one. But it's just scratching the surface. We've got the most common genetic variations, and do even have all of them. So at things above 5% in the population, we have a pretty darn good catalog. But from 5% to 0.5%, we're not doing so well on the catalog. And below that, it's rare genetic variants, so rare that you're going to have to get them by sequencing individual patients. And so I just want to say a moment, and this is for the aficionados in the audience, so forgive me, about the architecture, the genetic architecture of diseases. So some of the things that people worry about with common variants and their relation to disease is they say, well, any one of those variants doesn't seem to have a very big effect. That's true, and that would be a worry if what your goal was to assign everybody their own personalized risk prediction. But if your goal was to understand the biological pathways underlying disease, who cares? Because in fact, most of those genes, when you go in, you then manipulate them much more dramatically, you knock them down, have much bigger effects. And then interestingly, people said, and the total variation explained by those genes isn't so big. Well, in fact, the initial studies that was true, but what's been interesting is watching that grow over time with larger and larger sample sets. So for example, for schizophrenia, the first study with 7,000 samples for schizophrenia had about no significant results. The next study, though, had 20,000 and it had significant results. The next one with 34,000 has a lot of significant results. And very interestingly for the mathematical types, if you plot on an axis the sample size normalized to some constant n that's specific to each disease, you find that the number of loci that are being discovered falls on a log linear plot here, on a log-log plot of sample size against number of loci being discovered, meaning they're all kind of following a power law with roughly the same exponent, which is really interesting because it says there's a lot more to be discovered if we looked at larger numbers in the population. In fact, some statisticians have beautifully shown indirectly that there's tons more genes to be found because a number of statistical methods have now allowed people to estimate the total effect of those things that we can't individually identify, and they explain even more. So there's a lot of discovery, and the discovery has really got to now be followed up biologically. At the beginning, when there were just a couple of loci here or there, it didn't mean much. As there are a lot more and they fall into pathways, people are now beginning to do really rich biological experiments and therapeutic hypotheses. But of course, that's just the common. There's still a lot to be learned about the rare genetic variations. So far, there's only been a small handful of examples of rare genetic genes that have lots of rare genetic variations in them, but no commons. There are a few good examples of that. And this is going to require many thousands of samples being fully sequenced. The network is underway, and we've got, there's some initial discoveries that have come out of that, but how far this will go is still unclear. And then lastly, I'll mention maybe even all the common variants and all the rare variants won't explain everything. Maybe it will turn out that when you know all the genetic variants, you still haven't explained it. And this is really for the aficionados, and then I'll go on past that point quickly. But if you take Crohn's disease, and you take 74 genes that have been found related to Crohn's disease in particular, and you ask how much of the genetic variance has been explained by these 74, the answer is, in all the papers, 22% of the genetic variance has been explained so far. You should always ask, but people rarely do, what were the assumptions under that? It turns out the assumptions were that all the genes worked in an additive fashion with no nonlinear interactions. Maybe the problem's the assumption. What if it turned out those genes worked in 3 pathways, and the phenotype depended on the rate-limiting pathway out of the 3? Under that trivial change in assumption, those same things would explain 58% of the genetic variance. What it tells me, at least, is we should not be trying to measure the percent of variance we've explained of anything, because we can't do it without knowing what's the biology. What we really need to do is discover genes and do the biology of those genes. That's bad news for the people who want to give you personalized risk prediction to take home. It's good news for the people who want to do biology and understand how they might do therapeutics around the biology. The road ahead is going to involve tens of thousands of samples being done around the world and pooling data. All right, I'm just going to very quickly rip through in the last few minutes a little bit about cancer. I'm not going to say a lot about it, a little bit of human populations, and I'll stop here. So, cancer. I could give you lots more slides about cancer with examples, but I won't. I'll just kind of say, instead of inherited genetic variation, somatically acquired variation. Instead of comparing lots of people, affecteds and unaffecteds, tumors and normal tissue. And this was actually one of the origins of the Human Genome Project. Renato Del Becco wrote an influential article back in 1986 saying, we need the sequence of the human genome if we're going to study cancer. And sure enough, being able to take unbiased genomic approaches have taught us a great deal. Enormous therapeutic advances have come from the Human Genome Project. Drugs against BRAF mutations in skin cancer, PIK3CAs in clinical trials, EGFR mutations in lung cancer are standard of care. Drugs against EGFR are standard of care now in lung cancer. IDH1 mutations in glioma and now in a number of other tumors have given rise to a number of things in clinical trials. Whole new classes of genes involved in lineage survival, transcription factors underlying different types of specific lineage cancers. Translocations used to officially only matter in blood cancers. Now they clearly matter in solid cancers, etc. So this whole international project to sequence many cancer genomes has gotten underway, and it's really remarkable what comes out of it. I'll just say one story is multiple myeloma. Work we've been collaboratively involved in, where sequencing a wide variety of multiple myeloma samples has taken us from the point where we knew 3 genes involved in multiple myeloma, KRAS, NRAS, and P53, to a world now where there are 7 novel pathways here. Protein homeostasis, critical mutations in 40% of samples, NF-kappa B pathway, 25% of tumors. BRAF mutations present in 4% matters because now they're anti-BRAF drugs, etc. So there's this picture now where we have now about 4,000 samples it's grown to at the Broad, many more thousand samples elsewhere. I think it's now up to about 7,000 samples around the world, and this is clearly going to grow toward 20,000, 30,000 samples in the next year or two. There's clearly very different mutation rates, mutation patterns, but we're getting to the point where there will be comprehensive catalogs sometime in the next 3, 4, 5 years of the kinds of mutations that we see at any appreciable frequency. So the road ahead is going to involve a lot of that work, but that's only the start. We're going to need to then take those mutational classes and figure out what are their functional consequences. Given that you had a particular set of mutations, what things are now essential? What are the synthetic lethalities, the dependencies, the Achilles heels of cancers as a function of their mutations? And folks are working up high-throughput ways to approach those sorts of questions. I'm going to put an advertisement at the bottom that what we really need is to make real progress in cancer genomics is kind of an international alliance of cancer patients everywhere in the world where they can check a box that says, please contribute my information to a common project so that we can learn as rapidly as possible who's responding to what drugs under investigation. I think that kind of sharing will drive progress tremendously in cancer. And there's a lot of interest right now in trying to get such a project, and I hope Israel plays a central role in such work. Last quick topic is just for fun, and that's mapping recent human evolution. So not medical consequence, well, actually there is a medical consequence, but mostly just for fun. We've known that humans evolve. Not just that we evolved to become humans, but we're still changing in response to environmental pressure. But we didn't know an awful lot about it. What we knew from 2001 or so was the official story in the textbooks. Again, what I taught the freshmen at MIT is the human population comes from Africa. The human population schlepped out of Africa into the Middle East, into Europe, into Asia, and eventually to the Americas. And that's what happened, and populations came out and split and split and split. We knew some interesting things must have happened along the way, like some selection, but we couldn't give you any good examples other than selection by malaria and the brilliant insight of JBS Haldane that many blood disorders correlated with the distribution of malaria, and in particular things like sickle cell anemia. What's been wonderful in the last 10 years, as more and more data are getting collected on populations, is that story of just migration and split is definitely now turned into a much more complex and fun picture. It's migration and mixing. Populations clearly migrated, split, and remixed. India is clearly a mixture of two populations that came together from the north and the south. And as Craig has already referred to in talking about his own family tree, Neanderthal, and mine as well, Neanderthal there, on average we're about 4% Neanderthal. That's a really cool mixture. And so it's become a much richer picture of population spread and mixing. And then with regard to positive selection, well, we only had this story of JBS Haldane and the distribution of malaria and these blood disorders, and it says something. But he had to have some inspired guess. But it turns out the genome itself contains the signatures. It contains the answers of where selection occurred because there's a smoking gun of selection in the human genome. Pardis Sabeti worked out this beautiful smoking gun, which is if an allele rises from rare frequency to high frequency and it's just random drift, it takes a long time. And genetic recombination doesn't allow it to bring along its neighbors. But if a mutation rises from rare frequency to high frequency quickly, it brings along its neighbors. And that block that it brings along is a smoking gun of selection. And it's pointed to more than 300 regions of selection across the genome. New techniques have made it possible to narrow them down to tiny little regions, find what genes matter. And it's clear that evolution is not done with us in any respect. Lots of these loci harbor things related to different infectious diseases. We shouldn't be surprised about that. To skin color, we shouldn't be surprised about that. Homeostasis and metabolism, immune responses, sensory perception. Anyway, what have we learned in 10 years? A little bit. We've learned a little bit. Compared to what we're going to learn in this next decade with the students in this room and the students everywhere in the world, it's just a little, but it's pointed to how fruitful this is going to be and why this whole human genome business wasn't such a bad idea. It also taught us a bit about how to study biology. The idea that working together, collaborative projects, sharing data freely, mixing and matching those sorts of things. That idea has also stuck, and that has driven work in so many fields. So it's taught us both about the human genome and a little bit about how the human species can best make progress in learning about itself. Anyway, I want to acknowledge that the work I've described is the work of many people, collaborators, and others who have allowed me to borrow some slides to describe cool work, like Seik Kathresen's beautiful work on lipids, and to acknowledge them at the Broad Institute, and to say, and I think my co-recipients will feel the same way, that the proper acknowledgement slide for the human genome talks, for all of our talks, is this one. This is the product of a world working together. All of this work of the Human Genome Project is the work of people everywhere around the world just freely pouring data in, sharing ideas, and it's an enormous tribute. And so to this next generation who's going to pick up and run with this work, get back to the lab. Thank you.